Bismillahirrahmanirrahim. Here's Dr. Fozia Anjum presenting mutagenicity evaluation of medicinal plants collected from different localities of Pakistan. First of all, some introduction about the medicinal plants or current status of the uses of the medicinal plants. In the current era of the natural world, scientists have been documented experimental proofs of the medicinal plants having therapeutic potential, and these plants-based curative medicines having no side effects are getting popularity day by day, and the consumption of the herbal medicines have been continued and augmented generation after generation and supposed to be ancestors of valuable drugs, but their hazardous and lethal side effects have also been reported. Most of these phytotherapeutic products having bioactives of adverse effects are considered as useful and safe erroneously, and therefore systematic exploration of scientific evidences are authoritative to validate the consumption of the plants of therapeutic potential in traditional medicament and evaluated for their efficacy and safety by following the same protocol used for the innovative drug formulations. And these drug formulations could be directly or indirectly toxic to human body and producing allergenic reactions after antagonizing with chemical drug formulations. Therefore, the current project was planned to evaluate the therapeutic potential of the medicinal plants, not only therapeutic potential but also their toxicity towards the human health. For this purpose, different medicinal plants were collected from different localities of the Pakistan and then further subjected to pre-treatment and then extraction of the bioactive components by using ultrasonic waves extraction, and then after the extraction of the bioactive components, these bioactive components were further subjected to concentration and then lyophilization, and then after the concentration and lyophilization, these bioactive components were further subjected to evaluation for their therapeutic potential and for the evaluation of their toxicity towards human health, and for the evaluation of their therapeutic potential different activities was performed. These were antibacterial activity, antifungal activity, and biofilm inhibition activity, whereas toxicity was evaluated by hemolytic activity and mutagenicity. After the evaluation of their therapeutic and toxicity towards human health, then the results were obtained, and these are given here. It is clear in this slide that these bioactive components extracted from different medicinal plants. In this slide, it is clear that these bioactive components exhibited antimicrobial potential and maximum antibacterial activities was exhibited by bioactive components of the Azardiracta indica. Similarly, antifungal activity was performed by using these bioactive components of extracted from these medicinal plants, and maximum antifungal activity was found high by bioactive components of the Azardiracta indica, and these bioactive components were also evaluated for their biofilm inhibition activity. For this purpose, these bioactive components were used, and maximum biofilm inhibition or microbial growth inhibition activity was found high in case of the bioactive components of the Azardiracta indica, followed by Solanum nigrum, Cassia fistula, Nicotiana-N, and then Calatropis procera. It means that these bioactive components have potential to inhibit the growth of the microbes, so can be used as for their and can be used for their therapeutic potential in medicament. And these bioactive components were also evaluated for their toxicity, and for this purpose cytotoxicity activity was performed and mutagenicity activity was performed by following the ailments fluctuation test by using the Salmonella typhimurium TA98 and TA100, and the results are given here. It is clear in this slide that cytotoxicity was found higher in case of the bioactive components of the Nicotiana tobacum, and then followed by Cassia fistulas, bioactive components of the Cassia fistula, Solanum nigrum, Azardiracta indica, and then Calatropis procera. Whereas mutagenicity was found high in case of the bioactive components of the Nicotiana tobacum, and then followed by bioactive components of the Azardiracta indica. So in the last, it is concluded that these medicinal plants not only showed exhibited their medicinal potential but also having toxicity. Maximum toxicity was displayed by active components of the Nicotiana tobacum, which showed maximum cytotoxicity as well as mutagenicity, whereas Cassia fistula and Azardiracta indica showed toxicity to some extent. So therefore, the safety of their use in medication should be carefully evaluated before the formulation of the drugs. Thank you very much.